treating cancer with combinations of drugs can be more effective than using a single drug. However, figuring out the optimal combination of drugs and making sure that all of the drugs reach the right place can be challenging. To help address those challenges, MIT chemists have designed a bottlebrush-shaped nanoparticle that can be loaded with multiple drugs in ratios that can be easily controlled. Using these particles, the researchers were able to calculate and then deliver the optimal ratio of 3 cancer drugs used to treat multiple myeloma. In a study of mice, the researchers showed that nanoparticles carrying 3 drugs in the synergistic ratio they identified shrunk tumors much more than when the 3 drugs were given at the same ratio but untethered to a particle. This nanoparticle platform could potentially be deployed to deliver drug combinations against a variety of cancers. For several years, the research team has been working on polymer nanoparticles designed to carry multiple drugs. In the new study, the research team focused on a bottlebrush-shaped particle. To make the particles, drug molecules are inactivated by binding to polymer building blocks and then mixed together in a specific ratio for polymerization. This forms chains that extend from a central backbone, giving the molecule a bottlebrush-like structure with inactivated drugs, prodrugs, along the bottlebrush backbone. Cleavage of the linker that holds the drug to the backbone releases the active agent. In this study, the researchers first tested particles carrying just one drug, bortezomib, which is used to treat multiple myeloma, a cancer that affects a type of B cells known as plasma cells. Bortezomib is a proteasome inhibitor, a type of drug that prevents cancer cells from breaking down the excess proteins they produce. Accumulation of these proteins eventually causes the tumor cells to die. When bortezomib is given on its own, the drug tends to accumulate in red blood cells, which have high proteasome concentrations. However, when the researchers gave their bottlebrush prodrug version of the drug to mice, they found that the particles accumulated primarily in plasma cells because the bottlebrush structure protects the drug from being released right away, allowing it to circulate long enough to reach its target. Using the bottlebrush particles, the researchers were also able to analyze many different drug combinations to evaluate which were the most effective. Currently, researchers test potential drug combinations by exposing cancer cells in a lab dish to different concentrations of multiple drugs. But those results often don't translate to patients because each drug is distributed and absorbed differently inside the human body. The researcher explains that if we inject 3 drugs into the body, the likelihood that the correct ratio of those drugs will arrive at the cancer cell at the same time can be very low. The drugs have different properties that cause them to go to different places, and that hinders the translation of these identified synergistic drug ratios quite immensely. However, delivering all 3 drugs together in one particle could potentially overcome that obstacle and make it easier to deliver synergistic ratios. Because of the ease of creating bottlebrush particles with varying concentrations of drugs, the researchers were able to compare particles carrying different ratios of bortezomib and 2 other drugs used to treat multiple myeloma, an immunostimulatory drug called pomalidomide and dexamethasone, an anti-inflammatory drug. Exposing these particles to cancer cells in a lab dish revealed combinations that were synergistic, but these combinations were different from the synergistic ratios that had been identified using drugs not bound to the bottlebrush. In tests in 2 mouse models of multiple myeloma, the researchers found that 3 drug bottlebrushes with a synergistic ratio significantly inhibited tumor growth compared to the free drugs given at the same ratio and 2 mixes of 3 different single drug bottlebrushes. They also discovered that their bortezomib-only bottlebrushes were very effective at slowing tumor growth when given in higher doses. Although it is approved for blood cancers such as multiple myeloma, bortezomib has never been approved for solid tumors due to its limited therapeutic window and bioavailability. The team is also working on using these particles to deliver therapeutic antibodies along with drugs, as well as combining them with larger particles that could deliver messenger RNA along with drug molecules.